This investigation will explore what happens to the leaves of a celery stem when the colour of the water is changed. The testable question is what happens to celery when we change the colour of the water. My prediction is that the celery will develop tinted leaves the same colour as the coloured water. The materials used are a head of celery, scissors, measuring cups and measuring spoons, an additional spoon for mixing, red and blue food colouring, 3 plastic cups, water, and an apron to protect clothing in case of spills. First, use the scissors to cut 3 stalks of celery, taking care to ensure that each stalk is approximately the same size. Now place 1 celery stalk in each of the plastic cups and, using the measuring cups, fill each one with 1 cup of water. After that, add 1 teaspoon of red food colouring to one of the cups and 1 teaspoon of blue food colouring to another cup. Use the spoon to fully incorporate the food colouring. Set aside for 2 hours. Repeat steps 1 to 3 2 more times for a total of 3 tests. To ensure a fair test, it is important that the independent variable, the food colouring, is the only change in the investigation, and all other aspects of the investigation, including the amount of water, size of the celery stalk, and the time waited, are controlled as best as possible. The results from each test were fairly consistent. All the blue and red celery stalks have clearly blue or red tinted leaves after 2 hours. Samples were taken from each stalk for comparison. Some leaves appeared to have absorbed the colour more than others. The conclusion of this experiment is that when a stalk of celery is placed in coloured water for a period of 2 hours, the food colouring tints the leaves of the stalk. The scientific concept underpinning this investigation is how water is transported and used by plants. Using celery stalks, students can observe how water travels from the base of the plant through the xylem to the tips of the leaves, where it may then evaporate or be used by the plant to make energy through photosynthesis. In planning and conducting this investigation, students make and record observations using oral descriptions and simple digital recording methods, taking photos to record results. Students also work cooperatively as part of a group to brainstorm predictions and to share results based on guided investigation. Students are also guided to use safe practices when handling materials such as food colouring. This investigation may be extended to design and production, as students may choose to apply this method to other plants such as cut flowers to attempt to create a multitude of colour varieties. How does this investigation meet outcomes related to early stage 1 living world? This investigation meets outcomes related to early stage 1 living world, as students explore the characteristics, needs, and uses of living things, recognizing that living things such as celery need water, and how water is used by the plant. Moreover, comparisons can be made between celery and larger plants such as trees. Students can also observe, question, and collect data to communicate ideas, in that students make predictions about what they believe will happen to the celery when the colour of the water is changed, and students observe the results of the investigation as the leaves and xylem change colour.